Hello guys, our chapter is entitled Oxygen Toxicity and Free Radical Injury. You all know that oxygen is essential for life. It is the final acceptor of electrons in cellular respiration, and without it, the respiration cannot proceed, nor the production of ATP. But sometimes when the oxygen accepts a single electron, it can be transformed into a highly toxic free radical as well as reactive oxygen species, which can cause damage to protein, lipids, and DNA. Now you can imagine the consequences like the cell death and the diseases. In fact, there are many diseases associated with a free radical injury, like the ALS, the disease that the great scientist Stephen Hawking used to suffer from. It was named the motor neuron disease. The ischemia and reperfusion injury, it's when the tissues are deprived of oxygen for a long period of time and all of a sudden the blood supply returns to tissues. And instead of restoring the normal function of tissue, an oxidative damage will occur. The oxidative phosphorylation disease, also known as the mitochondrial DNA damage, the Alzheimer's disease, the Parkinson's disease, diabetes, and aging. You know, all these diseases will occur when the body defense mechanism, like the oxidant and the enzymes, fails to encounter the radical oxygen species and the reactive oxygen species and the reactive nitrogen oxygen species. So when there is a high amount of ROS and RNOS, even greater than the cell defense mechanism, it will lead to an oxidative stress, leading to a damage of lipid, proteins, and DNA, and finally tissue injury and inflammation. Now, you may be thinking that the reactive oxygen species are abnormal to occur, but no, this is not the case. They occur on a daily basis. They are formed both by enzymatic and non-enzymatic processes, in most cases as accidental byproducts. And in some cases, the immune cells, they produce reactive oxygen species to kill the pathogens. The UV radiation and the pollutants also produce reactive oxygen species. Now, these reactive oxygen species are formed from the oxygen, which is a bi-radical, that when it accepts electrons, it leads to the formation of reactive oxygen species, such as the O2 minus, the superoxide anion, which is a free radical and a reactive oxygen species. H2O2, the hydrogen peroxide, is a reactive oxygen species, but it's not a radical. The hydroxyl radical, which is a radical and a reactive oxygen species. These reactive oxygen species are dangerous because they can cause protein, lipid, and DNA damage, like I said, if they have not been neutralized by the antioxidants. Now, a radical by definition is a molecule that has a free electron in an orbital and it's not easily neutralized. Radicals are highly reactive. So what this means that they can extract electrons from other molecules to complete their orbitals. When they extract electrons from other molecules, they transform them into radicals. So what happens is when the oxygen accepts an electron from a coenzyme Q, for example, like in the electron transport chain, I'm going to talk about it in details later on. So when the oxygen accepts an electron from, for example, the coenzyme Q, it's transformed into a superoxide anion, which when it accepts another electron and two protons, it forms the hydrogen peroxide, which will accept another electron to form water and hydroxyl radical. The hydroxyl radical, when it accepts electron, it forms a water. So from oxygen to water, we need 4 electrons, 1, 2, 3, and 4 electrons. So the oxygen, when it can accept a total of 4 electrons to form water. Of course, I want to remind you here about the hydrogen peroxide. You used to know that from other courses that we can, the body has an enzyme that neutralizes this reactive oxygen species, and it's called the catalase. We're going to talk about it later on, but in brief, of course. Now, the highly reactive now the formation of reactive oxygen species like the hydroxyl radical can be done through two non-enzymatic reactions by a transfer of single electrons. These reactions are called the Haber-Wise reaction and the Fenton reaction. Let's see the first one. The Haber-Wise reaction involves an interaction between the superoxide anion and the hydrogen peroxide in the presence of proton to form oxygen, water, and the hydroxyl radical. Now, for the Fenton reaction, it involves transition metals, the Fe2+ and the cupric ion. So what happens in this case? The hydrogen peroxide is broken in the presence of ferrous or the cuprous, the Cu+, the cuprous, to form the hydroxyl radical and the hydroxyl ion, and of course the Fe3+, the ferric and the cupric ions. Now, this is the table that summarizes the most famous reactive oxygen species. The superoxide anion, the O2-, it's produced by the electron transport chains and other sites. It doesn't diffuse far, not like the hydrogen peroxide. It generates other reactive oxygen species, like you can see here from the superoxide, we have generated the hydroxyl radical, such as by the reaction of H2O2 in Haber-Wise reaction. Now, the hydrogen peroxide is a reactive oxygen species, but not a radical like I've already said. It generates free radicals by reaction with transition metals like Fe2+ and Cu+. It diffuses into and through cell membranes, not like the superoxide anion. So I'm going to remind you of the Fenton reaction. The hydrogen peroxide is split into hydroxyl radical and hydroxyl ion in the presence of transition metals. The hydroxyl radical is the most reactive in attacking biological molecules. And we said that we can obtain the hydroxyl radical by two non-enzymatic reactions. What are they? The Fenton reaction and the Haber-Wise reaction. Now, for the hypochlorous acid, this is a reactive oxygen species that's produced from H2O2, the hydrogen peroxide, by neutrophils to destroy invading organisms. Now we're going to talk about the major sources of reactive oxygen species, such as the coenzyme Q, the oxidase, oxygenase, peroxidase, and the ionizing radiations. Now we're going to start with the coenzyme Q generating peroxide. The reactive oxygen species are constantly being produced in the cells, with about 3 to 5% of the oxygen we consume being transformed into oxygen-free radicals. This means that when you consume oxygen, okay, 3 to 5% of this oxygen consumed is transformed to oxygen-free radical in the cell. One of the major sites of the production of superoxide is in the electron transport chain in the coenzyme Q, also known as ubiquinone. If you remember this molecule, we talked about it in the course of Biology 200 in the electron transport chain. This is the figure here show you the electron transport chain. This is the complex 1. This is the complex 2. This is Q, the coenzyme Q, or the ubiquinone, that receive electrons from the complex 1 and from the complex 2 and transfer them to the complex 3. Here, the same figure here represent also the complex 1, the coenzyme Q, and the complex 3 directly without showing the complex 2. So what happened in this case is that the ubiquinone is free within the membrane and can accidentally Okay, so it's free within the membrane and can accidentally transfer an electron to O2, forming the superoxide. Of course, when the O2 binds to the cytochrome oxidase, the complex 4, which is O2, you all know that's the final acceptor of electron. In this case, no radical oxygen, no reactive oxygen species occur in this at this level, because it's the normal function of the oxygen is to receive an electron and got transformed into water. But when the coenzyme Q accept an electron from the complex 1 and the complex 2 and got reduced, one of the electron can leak and pass to the oxygen, which can be transformed into the superoxide. Another source of reactive oxygen species are the enzymes oxidase, oxygenase, and peroxidase. Most of them can produce superoxide anions. How is that? They can bind oxygen, because they can bind oxygen and transfer an electron via a metals, leading to an accidental leakage of the free radical intermediates before the reduction into water is complete. So let's take an example, the cytochrome P450, also known as a monooxygenase. Usually, this is a protein, a heme protein, that's secreted in the body in response to drugs and alcohol, or when we are exposed to carbon tetrachloride, the CCl4, which is used as a solvent in the dry cleaning industry. The body in this case increases the synthesis of cytochrome P450, which main role is to detoxify these toxic compounds. But while doing that, they accidentally produce toxic reactive oxygen species. Not only the monooxygenase can form reactive oxygen species, but also the peroxidase, which are the enzymes involved in the degradation of the very long chain fatty acid in the peroxisome. While doing that, they produce the reactive oxygen species H2O2, the hydrogen peroxide, which is detoxified by the catalase enzyme, as I mentioned before. And all this occur in the peroxisome. The oxidase enzyme can also lead to the production of reactive oxygen species. Now, the exposure to cosmic rays, radioactive radiation, and X-rays, they can cause also the formation of reactive oxygen species, because they have high energy that can break down the water. Where's the mouse? Okay, so they can break down the water into hydroxyl radical, forming hydroxyl radical and hydrogen atom. So this will lead to a skin damage, mutation, and cancer. Like I said before, these reactive oxygen species are quite dangerous because they can interact and damage cell components, like you can see here in this figure. So the reactive oxygen species toxicity is due to the damage they can cause to the cell. They can damage the protein, the lipid, and the DNA. Let's see how. The superoxide anion and the hydroxyl radical can initiate membrane, mitochondrial, nucleus, and ER damage and peroxidation. So they can initiate the peroxidation, the lipid peroxidation in these compartments, leading to lipid damage and thus to membrane damage, because the membrane is formed of lipids. So what happens in this case? When there is a membrane damage, this will increase, or the membrane peroxidation, lipid peroxidation of the membrane, this will increase the permeability of the membrane. This will lead to an increased permeability to the membrane, to the ions and to water, leading to a high influx of calcium into the inside the cell. This influx of calcium will lead to mitochondrial damage and free radical production. Not only that, but it will also lead to an oxidation of the cysteine amino acid. You all know that the cysteine have a sulfhydryl group SH, and it's very important because two cysteine molecules can form SS bond and favorize the stabilization of the protein. But when they become oxidized, this will lead to a damage and degradation of the protein. Not only that, but the DNA also will be oxidized and damaged. So one of the effects of the free radicals that we have described in the previous slide is the lipid peroxidation. It can be initiated when a free radical like the hydrogen peroxide extracts an electron from the lipid, a polyunsaturated fatty acid, thus forming a lipid radical, and the hydroxyl radical itself becomes saturated. The second step is the propagation. The propagation of the chain reaction is done when the free lipid radical interact with the oxygen to form the lipid peroxy radical, which is then reduced to form the lipid peroxide. The third step is the degradation, in which there is a rearrangement of the electron in the lipid peroxide, which can result in the degradation of the lipid, forming two molecules, malonaldehyde and the degraded lipid peroxide. The malonaldehyde is a blood-soluble molecule, which is usually used as a marker for radical oxygen species injury. Now, the last step is the termination. To terminate this reaction chain, the body defense mechanism got involved. A potent antioxidant, the vitamin E, can terminate the chain of reaction by donating an electron. So the vitamin E is gonna donate an electron to the lipid radical, and in this way, the chain reaction terminates. But now the vitamin E is oxidized. It must be reduced again into this form to be able to carry this reaction again. This is done by the vitamin C. So the vitamin C restores the function or regenerate the vitamin E. The reactive oxygen species can also attack proteins, as I said before. The histidine, the arginine, cysteine, and methionine are the most susceptible amino acids in protein to hydroxyl attacks and oxidative damage. As a result of the attack of radical oxygen species, because they extract an electron from these proteins, the proteins got fragmented, or they can cross-link and will be degraded eventually. One of the important proteins is the tripeptide glutathione. It's made of 3 amino acids. This glutathione is also an antioxidant. It usually attacks the radical oxygen species in an attempt to neutralize them. But what could happen is that the reactive oxygen species can overcome the activity of the glutathione and oxidize it by oxidizing the cysteine amino acid in it. So the glutathione got fragmented and degraded. In this way, the cell loses a part of its defense mechanism, and this will lead to an accumulation of the reactive oxygen species. The DNA is also a major target of the free radicals attack, and there is at least 20 different types of modified DNA that has been reported. One of which is the attack of guanine that got converted in the presence of the hydroxyl radical to 8-hydroxyguanine. This 8-hydroxyguanine could be measured as an indication of the oxidative damage in the cell, in the DNA. And particularly, and it will also lead, the 8-hydroxyguanine could also lead to a mispair of the bases. But of course, you know that the DNA has a mechanism of repair in itself. The first part of this chapter was about the reactive oxygen species. Now we're going to talk about the reactive nitrogen oxygen species. Nitric oxide is a starting molecule to form the reactive nitrogen oxygen species, just like the oxygen was for the reactive oxygen species. Nitric oxide is also essential for the life, but can also be toxic in some cases. As a gas, it can diffuse into the cytosol and through membranes between and into cells. At low concentrations, nitric oxide acts as a neurotransmitter and a hormone that causes vasodilation of the heart. It increases the diameter of the blood vessels and thus it lowers the blood pressure. But at high concentration, that's when the nitric oxide become a problem because it combines with oxygen or superoxide to form the reactive nitrogen oxygen species, which are involved in several diseases. Nitric oxide exerts its effect in the target cell by binding to iron heme group in the enzyme, like the guanylyl cyclase. Do you remember when we talked in another course about the adenylyl cyclase, or the also known as the adenylate cyclase, and that it forms from ATP a cyclic AMP, the second messenger? Well, the enzyme guanylyl cyclase forms cyclic GMP, and also it activates a signaling cascade similar to the cyclic AMP. And since nitric oxide is a gas, it can rapidly be inactivated by bonding to other molecules. That's why it needs to function over a short distance from the source. So the nitric oxide at high concentration, it can be toxic because it can form reactive nitrogen oxygen species. You can see here that when the nitric oxide, okay, let me use annotation. Okay, so this is the when the nitric oxide is formed. Later we're gonna talk about the mechanism of synthesis of the nitric oxide. But when it's formed, it can combine with another nitric oxide and radical and oxygen to form later on the nitrogen trioxide. It can combine with the superoxide anion to form peroxynitride, and the peroxynitrous acid that can give rise to the formation of the nitronium ion and nitrogen dioxide with the hydroxyl radical. So these are the different forms of the reactive nitrogen oxygen species. And like the reactive oxygen species, the reactive nitrogen oxygen species can cause damage to protein, to lipids, and to DNA. And it can cause neurodegenerative diseases like Parkinson and the rheumatoid arthritis as a chronic inflammatory disease. So now we're going to talk about the formation of the nitric oxide. Nitric oxide is synthesized from the conversion of arginine into citrulline through the nitric oxide synthase. This enzyme required NADPH and oxygen to form the nitric oxide. There are three isoforms of this enzyme, the nitric oxide synthase. We have the neuronal nitric oxide synthase, the endothelial nitric oxide synthase, and the inducible nitric oxide synthase. The neuronal exists in the neurons, the endothelial in the endothelial cells, and the inducible exists in immune cells. Now, these two are regulated by the calcium. So a low amount of nitric oxide is required for its role as a hormone and a neurotransmitter, whereas the inducible, which exists in immune cells, is regulated at the gene level. The inducible, also you have to know that the inducible nitric oxide synthase produce high level of nitric oxide to help in the destruction of invading microorganisms. Now, when there is a microbial invasion, phagocytic cells will do a respiratory burst, which is described as a rapid consumption of oxygen to form reactive oxygen species and reactive nitrogen oxide species that can destroy the invading pathogen or even the tumor cell. So what happens when an immune cell like neutrophil encounter a microbe? It engulfs it. You see how does it engulf it? Okay, so it engulfs the microbe and recruit NADPH to its membrane, to the membrane of the immune cell, where it donates. So this NADPH is gonna donate an electron to oxygen to form the superoxide anion. This is done by the action of the enzyme NADPH oxidase. So do you remember when we said before that oxidase are a source of the reactive oxidant species? This is an example where NADPH oxidase forms the superoxide anion. Now, the superoxide anion can form the hydrogen peroxide. It's converted to hydrogen peroxide. The hydrogen peroxide will combine with the chloride catalyzed by the enzyme myeloperoxidase to form the hypochlorous acid. This one is very important and a very powerful reactive oxygen species because they can destroy the bacteria within seconds of interaction. Now, the hydrogen peroxide can also generate the hydroxyl radical via the Fenton reaction that involves the transition metal Fe2+. So the H2O2 can also generate the hydroxyl radical from the Fenton reaction and can damage the bacteria. The inducible nitric oxide synthase may be activated and generate nitric oxide that can combine with this one. The nitric oxide will gonna combine with the superoxide to form the peroxynitrite, which is going to destroy the bacteria. This process is called the respiratory burst because it takes only 30 to 60 minutes and it consumes oxygen. Now, after explaining the reactive oxygen species and the reactive nitrogen oxygen species, their formation and their effect, we need to talk about the defense mechanisms that the cell have against the oxygen toxicity. One of the cellular mechanisms used to fight against the oxygen toxicity is the antioxidant scavenging enzymes, like the superoxide dismutase, like glutathione peroxidase and glutathione reductase, the presence of the catalase enzyme, and many others. We have also the non-enzymatic antioxidants that are called the free radical scavengers, like the vitamin E, like the vitamin C, like the beta-carotene and the flavonoids. We have another mechanism that can be used to fight against the oxygen toxicity is the compartmentalization of the reactive oxygen species, which help to keep the reactive oxygen species in vesicles away from the critical proteins and the critical sites. Another way is the metal sequestration. Because you know that Fe2+ and Cu+ are needed for the Fenton reaction. So sequestering or quenching the metals with special proteins such as ferritin help in this process because these metals will no longer be free and available to undergo the Fenton reaction. And this is how we can prevent the formation of reactive oxygen species. We have also the mechanism of repair damage components. For example, the chaperones, if the protein are misfolded, and there is the DNA repair mechanism if the DNA is damaged. Now we're going to talk about the first mechanism that the cell have to defend or to fight against the oxygen species, which is the antioxidant scavenging enzymes. Here I'm going to mention the superoxide dismutase, abbreviated SOD. You can see here how the superoxide dismutase converts the O2 minus the superoxide anion to hydrogen peroxide. You have to know that the superoxide dismutase exists in 3 isoforms. One exists in the cytosol, another one in the mitochondria, and in the extracellular medium. Now we're going to say how is the conversion of a radical oxidant species into another radical oxidant species an action of scavenging. Well, it's because it converted into a one that the cell have already an enzyme to to neutralize it, which is a catalase. The hydrogen peroxide is then reduced by the catalase into water and oxygen. So this mechanism, this enzyme that exists in the peroxisome, prevent the hydroxyl formation from the hydrogen peroxide. Another enzyme, the glutathione peroxidase and the glutathione reductase. Do you remember when we talked about the glutathione, the tripeptide? When we talked about it, we mentioned that it was an antioxidant molecule formed of 3 amino acids, and the cysteine was very important. Well, how does glutathione act? In fact, 2 molecules of glutathione will combine together under the action of glutathione peroxidase to form the glutathione disulfide, GSSG. The SH here is that of the cysteine, and you all know that the sulfide dry group, when 2 sulfide dry group exist close to each other, they're gonna form an SS bridge. So the glutathione, 2 glutathione molecules are condensed, and there is a formation of glutathione disulfide under the action of glutathione peroxidase. Now we're gonna ask, where does the 2 hydrogen goes? They goes to reduce the hydrogen peroxide. It's not just the hydrogen peroxide, sorry. It's not just the hydrogen peroxide, but every toxic molecule. It could be a lipid peroxide or a lipid radical. So the function of glutathione, because it's an antioxidant, it acts by reducing the toxicity, or they detoxify the toxic molecule and the reactive oxidant species such as the hydrogen peroxide. So they're gonna donate the hydrogen, the 2 hydrogen, to the hydrogen peroxide to reduce it into water, and in this way it becomes oxidized. Now, in order to be able, the glutathione to be able to do this over and over again, we must regenerate the glutathione in its reduced state. To do that, so to regenerate GSH, a molecule, we need the action of another enzyme called the glutathione reductase. This enzyme requires a coenzyme called NADPH that comes from the pentose phosphate pathway. So this is what I need you to know about the glutathione peroxidase and the glutathione reductase involved in the mechanism of defense against the reactive oxygen species. Now we're going to talk about the non-enzymatic antioxidants, starting with the vitamin E, also known as the alpha tocopherol. The vitamin E is an antioxidant. It's a lipid soluble. It protects against the lipid peroxidation in membrane. Of course, you remember when we talked about the lipid peroxidation and how the vitamin E was involved in the termination step of the lipid peroxidation mechanism. So we say that the vitamin E is a non-enzymatic terminator of free radical chain reaction. It exists in vegetable oils, liver, cereals, and others. Now, another non-enzymatic antioxidant or free radical scavenger is the vitamin E, also known as ascorbic acid or the ascorbate. The vitamin C. We talked about the vitamin C when we said that the vitamin E must be regenerated in order to complete the reaction of terminating the free radical chain lipid peroxidation. And I said previously that the vitamin C is the one that can help to regenerate the vitamin E, because it can donate an electron to the vitamin E. Vitamin E is a lipid soluble. That's why it needs to be transported via lipoprotein in blood. Whereas the vitamin C is a water-soluble molecule. It circulates in blood and fluid to access the membrane. It's also a redox coenzyme for the collagen synthesis. Now, the carotenoids or the beta-carotene, which is a precursor of vitamin A, it's also an antioxidant found in fruits and vegetables. It may slow down cancer and arteriosclerosis. Lutein and zeaxanthin that exist in the macula of the eye, they may help protect against the macular degeneration of the retina. Another exogenous antioxidant are the flavonoids. So here I mentioned a term exogenous because the previous one, antioxidant we talked about, like the vitamin C, the vitamin E, the carotenoids, and the flavonoids, they are exogenous because we can obtain them from the diet or as a supplement. And I need to refer to this as this term exogenous because we have also endogenous antioxidants. So let's focus now on the flavonoids, which are exogenous antioxidants, like for example the resveratrol that exists in the grapes. They may inhibit enzymes responsible for the reactive oxidant species, like the xanthan oxidase, and they may chelate the iron and the copper metals used for the Fenton reaction, and in this way they act as a free radical scavengers. Now, finally, we're going to talk about the endogenous antioxidants like the melatonin and the uric acid. Uric acid is the one that causes inflammation here and deposit on this location, and usually it's due to a huge consumption of protein. But uric acid in a normal amount, synthesized in a normal amount in our body, is helpful. It's an antioxidant. The melatonin is the one used to control the sleep cycle, the sleep awake cycle. It's also an antioxidant. And now we've reached the end of this chapter. Thank you for your attention, guys.